This is part 4 of Lecture 11 on Respiratory Physiology. This information can be found in Chapter 22 of your textbook on pages 819 to 857. Control of respiration. Respiratory rhythms are regulated by higher brain centers, chemoreceptors, and other reflexes. The neural controls involve neurons and the reticular formation of the medulla and pons of your central nervous system. The medullary respiratory centers are a cluster of neurons in two areas of the medulla are most important. There is the ventral respiratory group and the dorsal respiratory group. The ventral respiratory group is a rhythm-generating and an integrative center. It consists of a network of neurons in the brainstem that extends from the spinal cord to the pons medulla junction and sets the normal respiratory rate and rhythm of approximately 12 to 15 breaths per minute. The inspiratory neurons excite inspiratory muscles via the phrenic nerve to the diaphragm and the intercostal nerves to the external intercostals. And the expiratory neurons inhibit expiratory neurons inhibit inspiratory neurons. The dorsal respiratory group, or the DRG, is a network of neurons located near the root of cranial nerve 9. The group integrates input from peripheral stretch and chemoreceptors, then sends information to the ventral respiratory group, or VRG, neurons. Much is not known about this group of neurons. The pontine respiratory centers. The neurons in this center influence and modify activity in the VRG. They act to smooth out transition between inspiration and expiration, and vice versa. They transmit impulses to the VRG that modify and fine-tune breathing rhythms during vocalization, sleep, and exercise. Lesions in this area of the brain can lead to apneutic breathing, where a patient takes prolonged inspirations. The generation of respiratory rhythm and the origin of breathing rhythm is not yet fully understood. One hypothesis is that there are pacemaker neurons in the VRG that control intrinsic rhythmicity. The most widely accepted hypothesis is that reciprocal inhibition of the two sets of interconnected pacemaker neurons in the medulla generates a rhythm. Each neuron set controls the other set to ensure a rhythm. Depth is determined by how actively respiratory center stimulates the respiratory muscles. The greater the stimulation, the greater the number of motor units excited, which leads to an increased depth of inspiration. Rate is determined by how long each center is active, and both are modified by changing body demands. Respiratory centers are affected by chemical factors, the influence of higher brain centers, pulmonary irritant reflexes, and inflation reflex. In terms of chemical factors, the most important of all factors affecting depth and rate of inspiration are changing levels of partial pressure of carbon dioxide, partial pressure of oxygen, and pH. The levels of these chemicals are sensed by central chemoreceptors that are located throughout the brainstem and peripheral chemoreceptors found in the aortic arch and carotid arteries. In terms of the influence of partial pressure of carbon dioxide, this is the most potent and most closely controlled. If blood partial pressure of carbon dioxide levels rise, known as hypercapnia, carbon dioxide accumulates in the brain and joins with water to become carbonic acid. Carbonic acid dissociates, releasing hydrogen ions, causing a drop in pH or increased acidity. Increased hydrogen ions stimulates the central chemoreceptors of the brainstem, which synapses with the respiratory regulatory centers. The respiratory centers increase the depth and rate of breathing, which act to lower blood partial pressure of carbon dioxide, and pH rises to normal levels. If blood partial pressure of carbon dioxide levels decrease, respiration becomes slow and shallow. Apnea is a breathing cessation that may occur when partial pressure levels of carbon dioxide drop abnormally low. Swimmers sometimes voluntarily hyperventilate to enable them to hold their breath longer. It causes a drop in the partial pressure of carbon dioxide, which causes a delay in respiration as partial pressure of carbon dioxide levels need to build back up. It can, however, cause dangerous drops in partial pressure of oxygen levels. The influence of the partial pressure of oxygen. Peripheral chemoreceptors in the aortic and carotid bodies sense arterial oxygen levels. Declining partial pressure of oxygen normally has only a slight effect on ventilation because of huge oxygen reservoir bound to hemoglobin. It requires a substantial drop in arterial partial pressure of oxygen, below 60 millimeters of mercury, to stimulate increased ventilation. And when excited, the chemoreceptors cause respiratory centers to increase ventilation. The influence of arterial pH. pH can modify respiratory rate and rhythm, even if carbon dioxide and oxygen levels are normal. It is mediated by peripheral chemoreceptors. Decreased pH may reflect carbon dioxide retention, accumulation of lactic acid, or excess ketone bodies. The respiratory system controls attempts to raise pH by increasing respiratory rate and depth. Factors influencing breathing rate and depth. Pulmonary irritant reflexes. The receptors in the bronchioles respond to irritants such as dust, accumulated mucus, or noxious fumes. The receptors communicate with the respiratory centers via the vagal nerve afferents and promote reflexive constriction of air passages. Same irritant triggers a cough in the trachea or bronchiole or a sneeze in the nasal cavity. The inflation reflex. The Herring-Brewer reflex. Stretch receptors in the pleurae and airways are stimulated by lung inflation. They send inhibitory signals to the medullary respiratory centers to end inhalation and allow expiration, and may act as protective mechanism more than as a normal regulatory mechanism. With exercise, adjustments are for both intensity and duration that need to be made. Hypernemia is increased ventilation in response to metabolic needs, and ventilation can increase 10 to 20 times. Ventilation increases abruptly, increases gradually, and then reaches a steady state. When exercise stops, there is a small, abrupt decline in ventilation, followed by a gradual decrease. The partial pressure of carbon dioxide, the partial pressure of oxygen, and pH remain surprisingly constant during exercise. An increased ventilation with exercise involves three neural factors. First, the psychological stimuli, the anticipation of exercise. Second, the simultaneous cortical motor activation of skeletal muscles and respiratory centers, that is, the muscles moving. Third is excitatory impulses to respiratory centers from proprioceptors in moving muscles, tendons, and joints. We will see a gradual increase in plateau of respiration that is caused by an increased rate of carbon dioxide delivery to the lungs. And this is the end of part 4 of lecture 11 on pulmonary physiology.